Hey everyone, in this video I am going to talk about VDJ recombination, but before we get into that I want to give some background information on the immune system. Well, I'm going to try and give some background information on the immune system. I know very little about the immune system. I know it is extremely complex, but it's really fascinating, and I love the molecular biology aspects, especially the molecular biology aspects of antibody production, which I think it's really interesting. But before we get to that, I just want to briefly touch on some aspects of the immune system so we can have some context to our discussion of antibody production. So we can divide the, say, mammalian immune system, or we're going to try and focus on humans, but the human immune system, into the innate immune system and the adaptive immune system. So there are many different types of cells in the innate immune system, and these cells tend to use these receptors called PRRs for pattern recognition receptors. And these receptors are encoded by the germline. Well, all of our genes are encoded by the germline, right? But what you're going to see is that these don't need any rearrangement, any recombination in order to function. Now, let me just touch on the definition of recombination. Now, so rearrangement of genetic material. So sometimes when we talk about recombination, we're referring to crossing over during meiosis. Sometimes we're talking about template mediated repair during DNA repair. And sometimes we're just talking about the rearrangement of genetic material. So with respect to this discussion, we are focusing in on that just simple definition of recombination as rearrangement of genetic material. So with the innate immune system, the PRRs, now these are just encoded by genes in our genome. No rearrangement is necessary for these to be expressed. And these recognize things researchers have termed PAMPs and DAMPs, so relatively clever names here. So PAMPs are pathogen-associated molecular patterns. And my understanding is these are things that, like certain proteins that are found in many different pathogens, but they're very similar. I think maybe like the flagellum of a bacterium. And so there's no need to adapt to these things. Most bacteria have it and it's unchanging. So we can use things that are encoded by the germline, such as PRRs, to recognize these. DAMPs, these are things that are associated with damaged cells. So it allows cells of our immune system to notice when some of our cells are damaged. So PRRs, these are essentially passed from parents to offspring unchanged, barring mutation during replication or something like that. PRRs can be passed from parents to offspring unchanged. Now, the adaptive immune system, the main cells involved here, and I didn't go over the many different types of cells in the innate immune system, but the cells in the adaptive immune system, those are going to be the T cells and B cells. I know very little about T cells. B cells, however, these are going to be our antibody producers. And this is what I'm going to focus on in our course. So these here, though, use recombination to adapt to new antigens. And antigens are the things recognized by the antibodies, and they're produced by, pathogens. So they're parts of proteins of pathogens. And I think maybe they can, I don't know if they're always proteins, but they can possibly be other things. I'm used to thinking of them as antigens, but I guess maybe certain sugars possibly could be antigens. Don't hold me to that. But the antigens do come from pathogens, and they're recognized by the antibodies. So now when let's say T cells or B cells, their genes, the receptors, or in case of B cells, it would be antibodies or immunoglobulins. Now, when they undergo rearrangements to form an immunoglobulin that recognizes the specific antigen, that rearranged gene cannot be passed to offspring. So that's a major difference between how the innate immune system passes genetic information to offspring and how the adaptive immune system really can't pass certain information to offspring. Okay, so I want to take a look at one receptor. So the B cell receptor. And there are many different types of receptors that bind antibodies. And I'll just focus on the B cell receptor because it's important in B cell maturation. So here's the plasma membrane. And what I'm going to put down here are these transmembrane proteins. And this is the B cell. This is the plasma membrane of the B cell. And these transmembrane proteins are going to be called Ig alpha and Ig beta. These are also called CD79A and CD79B. And they have these important domains, which I'm just going to call ITAM, ITAM domains. And we'll talk about those in a second. Now, these right here interact with antibodies in the membrane. So antibodies, and here's just a rough structure of an antibody. Now, antibodies are going to have two heavy chains. Well, your typical antibody, we're going to call this a monomer because sometimes there are some antibodies that you can get multiple monomers linked together to form more complex antibodies. But here's your typical antibody structure right here. And these antibodies right here, I'm going to go into more detail on the antibody structure in a second, but this will interact with Ig alpha and Ig beta. And when these bind an antigen, say an antigen from a pathogen, it will trigger signaling by phosphorylation of the ITAM domains. So the ITAM domains of these proteins here will be phosphorylated, and then that will trigger signaling that will activate other defense processes. Activates various defense processes, whatever the next stage of the defense process will be. So this is a B cell receptor. And now I want to focus in on the antibody structure. So a fancier name for an antibody is an immunoglobulin. Abbreviated Ig. So they're the same thing. And I'm going to put a little bit more detail in this diagram here. So I'm going to put 2 heavy chains here. So there's 1 heavy chain, and see that squiggly line? Now these are identical. I drew this one longer, I didn't mean to. These should be identical proteins right here. And this is the hinge region of the heavy chain. And then the light chain is usually diagrammed out here. So this is 1 light chain. So light chain and heavy chain. So the other heavy chain is here, the other light chain is there. Now I'm going to shade in segments of the heavy chain and light chain. So the shaded sections are going to be the constant regions of the heavy chains and light chains. So constant regions. The N-terminal ends of the proteins are up here, the C-terminals are down here. Now, the unshaded parts, these are the variable regions, also called the FAB. F is for fragment, AB is for antibody binding. So the variable regions are the parts that are responsible for binding the antibodies. Did I say antibodies? I meant antigens. So the variable regions are the parts that are responsible for antigen binding. So AB is for antigen binding. So in this part over here can bind an antigen, and this part over here can bind the same antigen. I used to think of these as these sides together would bind an antigen, but I believe it's more correct to think of it as this can bind the antigen, and this can also bind the similar antigen independently of each other. I should also say the light chains and heavy chains are held together by disulfide bridges. We don't need to worry too much about that. I'm sure you will cover those in biochemistry. But that's what holds the heavy chains together with the light chains, disulfide bridges. And I think there are some up here too. So the constant regions are also called FC regions, and there are other receptors that bind antibodies, and those would be, some of those are called FCRs. So FC receptors, based on the fact that they bind the FC, the constant regions of the antibodies. Now, remember how in previous lectures we talked about epitopes, and how epitopes are the part of an antigen that is bound by an antibody. So these variable regions here, they bind the epitopes. And the very specific parts of the variable regions that bind the epitopes, those are called paratopes. So when you get really down to the nitty-gritty of the interaction, it's the paratope of the variable region binds the epitope of the antigen. And that's how this recognition takes place. Now, we're going to spend most of our time over the next lecture, maybe 2 lectures, talking about how cells form the genes. I guess how cells rearrange the genes that are responsible for producing the heavy chains and the light chains, and how those changes allow cells to recognize new antigens and new pathogens. So in humans, we have 2 genes for light chains. And these are very strange genes. They're genes that are made up of gene segments. So they're very strange to think about and hard to define. So let me give you an example, or one example. So the IgK locus, where K is for kappa. I may sometimes call it IgK, I may sometimes call it Ig kappa. Now, this is found on chromosome 2. So every human has 2 IgK loci. And at the IgK locus is responsible for producing one of the light chains, or a light chain. Now, humans also have another locus that can make a light chain. That's called IgL. So the IgL locus, which is on chromosome 22. And this is called the lambda locus. So this is also responsible for producing a light chain. And as we'll see, I think maybe tomorrow, maybe Wednesday's lecture, or in the next lecture, that each B cell is only going to make either a kappa light chain or a lambda light chain. It won't make both. There's a process called allelic exclusion, which will make sure that each B cell only makes one of these, either a kappa or a lambda. Now, there's one locus in humans that is responsible for producing the heavy chain. It's the IgH locus. IgH locus, so responsible for the heavy chain. I already wrote heavy up here. So the IgH locus, and that is found on chromosome 14. So each human has 2 IgK loci, 2 IgL loci, right? Because we have 2 chromosome 2s and 2 chromosome 22s, and we also have 2 IgH loci because we have 2 chromosome 14s. Now, these loci are made up of segments, gene segments, which is something we haven't really seen before, and I don't really know of any other examples of genes that are made up into these things that we call gene segments. And the segments must be rearranged before these genes can be transcribed into a functional protein. So I'm going to cheat a little bit here, speed things up. Let's just take a look at, well, I should say, even in the lecture notes, what I did was we're only going to look at IgK because IgL is very similar. So the rearrangements that happen at IgL very similar to IgK. So we're going to take a look at the IgK locus and the IgH locus. So the rearrangements that occur at both of these to make a light chain and to make a heavy chain. So now the process that rearranges these loci is called VDJ recombination. Now the V is for variable, the D is for, I'm forgetting what the D is for. The J is for joining, and the D is for, I can't believe I forgot what that is. I'm really embarrassed. D is for, of course my computer logged me out. And I forgot my password. Diversity. I came up with it before I saw it in my notes. I'm just going to check. Yes, diversity. I can't believe I forgot that. How embarrassing. So, the IgK locus contains variable gene segments and joining gene segments. The IgH locus contains variable segments, diversity segments, and joining segments. So I'm going to go over that in a moment again anyways. Essentially, IgK and IgL, which are very similar, don't have these diversity segments. They all have constant segments also that aren't really involved in this VDJ recombination process. And we're going to talk more about the constant regions in either I think in 2 lectures. So how does VDJ recombination work? So let's just take a look at the IgK locus in humans. So, like I said, these loci are very complicated, and it's hard to find a reference multiple references that agree on the number of variable gene segments. So these are going to be the variable gene segments down here in red. These are going to be the joining gene segments in yellow, and this is the constant gene segment. The IgK locus only has 1 constant segment, so that's pretty easy to remember. Now, references generally agree there are 1, 2, 3, 4, 5 joining segments at the human IgK locus, but the number of variable gene segments are, numbers range from 40 to about 300. And different individuals may have different numbers of these. So what we're going to say is, okay, there are dozens of variable gene segments. Now, I've shaded the gene segments red for variable, yellow for joining, and blue for constant. And I put a little K here to indicate that we're talking about the segments at the IgK locus. I could put constant here, and I've numbered these down here. So I numbered them backwards. So this is 1, 2, 3, 4, and we could go to whatever the last one is. I put this little hatch mark here to indicate that there are many more. If we should go upstream here, and I numbered the joining 1, 2, 3, 4, 5. And these numbers are going to be important because it's going to help us see what happens during the recombination, VDJ recombination at the IgK locus. Now, I should also say, because the IgK and IgL loci don't have diversity gene segments, that sometimes researchers refer to this as VJ recombination, and they leave out the D. But we're going to refer to all recombination involving variable and joining and diversity, if it's there, as VDJ recombination. If we call it that, we're okay. You're going to get that answer correct on the quiz. So, and right here, I'm just specifying V to J recombination at this particular point in the mechanism, because what I'm going to show you is that the very first step of VDJ recombination at the IgK locus is one of the variable segments is joined to one of the joining segments, and this joining is mostly a random process, so we can get any variable gene segment linked up to any joining gene segment. And there are some advanced studies which say some joining events happen more than, some specific joining events happen more than others, but for our purposes, let's consider them to be completely random. So we could link up 1 with 1 or 1 with 4, or we can link up 3 with 2, or and so on and so forth. So down here, what I'm showing you is, okay, so what do we do? So in this example, we're saying the cell picked somehow chose to link up number 3 right here with number 3 down here. And you can see how we did that here. So 3 is now fused to number 3. Everything between this point right here all the way over to before this point right here, all of that is cut out and lost. So this is lost, this is lost, this is lost, this is lost. All the intergenic sequences between the gene segments, all that is going to be thrown away by the cell. So after the V to K joining, after the V to J recombination event, we've lost all of this, and now the locus looks like this down here. So we have number 4 right here, and then 5, 6, 7, 8, however many more there are, they will all still be there in the genome. As you can see, 4 and 5 are still there. We lost the number 1 and 2 joining segments. 4 and 5 are still there, but that's not going to interfere with production of the light chain. So each of the variable gene segments has its own promoter. So all whatever 100 of these things or more, they all have their own promoter, but only one promoter works. It's the promoter that is closest to the joining gene segments. So even though there's a promoter here, it's not going to work. Only the promoter that is closest to the joining gene segments will work. So after this recombination event here, RNA polymerase will bind to this promoter down here, and it will transcribe the IGK locus, starting here and transcribing all of these down here. So we're going to end up with this, I guess, pre-messenger RNA that still needs to undergo processing. Now, I'm sure you've seen transcription in introductory biology and our introductory genetics course. So we're going to talk more about transcription in the future. But for now, this is the first transcript that's made. This is the pre-messenger RNA, and it needs to be processed by splicing, which means removing the introns, right, joining the exons. We need to add a 5' cap, which is going to be the 7-methylguanosine cap, and a poly-A tail. So what we're mostly interested here is the splicing that happens. So essentially, we have this variable gene segment right here that was fused to this joining gene segment. Now, there's going to be a 5' splice site right here. So everything after this joining gene segment to the very beginning of this constant gene segment is going to be removed by the spliceosome, the machinery that removes introns. So everything between here, 4 and 5 right here, and all these little pieces right here, they're going to be cut out of the transcript. So same thing would have happened if this, say, number 3 was fused to 4. Well, then this would have been cut out during the splicing event. So essentially, the 5' splice site that is used for removing all this stuff right here is always found after the first joining segment, 5' splice site, and this is always the 3' splice site. So after this transcript is processed, where'd it go? Okay. We're going to have a messenger RNA that looks like this. So this is the messenger RNA that is from the IGK locus in that one B cell that underwent that specific VDJ recombination. So after splicing, those other joining segments are gone. And now this could be translated, the start codon up here, stop codon down here, and that's going to give us our light chain right here. So these are the variable regions right here. This is the constant region down here. So at this point, you should see how we can make many different types of light chains depending on, which of these variable regions were fused to which of these J regions down here. And we are going to talk more about the specific details of how this recombination process works and how we can get some mutations put in here or add some more variation in addition to just joining different variable regions to different joining regions, or different variable segments to different joining segments. And we'll see that in the next lecture. Right now, we're just focusing in on joining, just the overall process of joining the different segments together. And we just have to now take a look at the IGH locus. Now remember, that was VDJ recombination at the IGK locus. or loci, because there are two of them. VDJ recombination at the IGL locus or loci is similar to IGK, so we don't need to talk about that. And why is it similar? Well, the IGL locus does not have no diversity segments. No diversity segments to worry about. Now, because the IGH locus has the diversity segments, it's going to be a two-step recombination. So here's my diagram of the IGH locus in humans here. Now, again, it's hard to get references to know how many, to give precise numbers that agree, multiple references that agree on the number of VH segments, so variable gene segments at the IGH locus in various humans. A recent study I just looked at said, different humans have different numbers of these things. Which, yeah, that makes sense. I don't know why I didn't predict that was probably going to be possible or even likely. So in my diagram, I have just dozens more. Let's just focus on there are dozens and dozens of these things at the IGH locus in humans. So here's my diagram of the IGH locus. The variable gene segments are over here in red. The diversity segments, now there are about 25 of these at the IGH locus, and these are in green. I've numbered them 1, 2 to 25 right here, and puts this little hatch mark in here to indicate that I haven't diagrammed all the various DH segments, because I'd have to make it really small and that would take forever, and it's not necessary to diagram all of them to understand what's going on. Now, most references agree that there are 6 joining gene segments at the IGH locus, and there are many constant regions. And as we're going to see, with respect to immunoglobulins, and maybe some of you already know this, that immunoglobulins come in many different types. Well, I think 6 major types, like IgM, IgD, IgG, IgE, etc., and they all have slightly different functions. So these will all use the same variable regions, but the constant regions change, and the constant region gives us the specific class, and these are going to be called isotypes. The type is determined by the constant region, and we're going to talk about how we get the different isotypes, I think, in 2 lectures from now. So I need another blank piece of paper. Okay. So again, here is, let me make sure I'm on screen still. Here is the IGH locus. Now, the first step to rearrangement of the IGH locus is going to be DH to JH recombination. So essentially, one of the cells B cell is going to choose one of these right here to join to one of these over here. Now, in my example, what did I do? I joined number 2 to number 4. So everything downstream of 2 and upstream of 4 was lost by the recombination event. So the cell discarded everything between those 2. And like I said, in the next lecture, we're going to see precisely how these are like fused together so perfectly. So in the next step, we're going to have VH to DH recombination. So the cell is going to link up one of these, going to choose one of the VH gene segments, whether it's number 1, 2, 3, or 80 or 100, however many there are. It's going to choose one of them to fuse to one of the D segments here. So in my example, I am fusing number 2 to number 2. So everything downstream of 2, this 2 right here, this VH number 2 right here, and upstream of this DH number 2 is going to be lost. And so after the DH-GH recombination and the VH-DH recombination, the IGH locus is going to look like this. So there are more constant CH segments down this way, and there are dozens more VH gene segments that way. But because we have our 2 recombination events, everything we need for formation of the heavy chain is going to be right here. So just like with the IGK and IGL loci, each of these variable gene segments has its own promoter. But the only promoter that is active, that is used by RNA polymerase, is going to be the one that's closest to the D and V segments. So this promoter right here in front of number 2 is the only one that's active or used. So the promoters in front of all the other gene segments, those aren't going to be used by RNA polymerase. So this is the promoter that's used. It transcribes a pre-messenger RNA that looks like this. And we can have 2 splicing events on this pre-messenger RNA. We can have this number 4 right here joined to the C delta constant region, or number 4 can be joined to the C mu constant region. And this is one model of how this happens. There's another model that explains how we get the different constant regions in the immunoglobulin or in the heavy chain. But one model suggests that, okay, the splicing event goes from 4 to the beginning of C mu. Another one says sometimes, same model, sometimes it goes to C mu, sometimes the splicing event goes to C delta. And that's going to give us 2 different heavy chains. But the most important part is everything downstream of 4 in between one of the constant regions is going to be lost. Whether it be 5, 6, and 5 and 6 that is lost, or 5, 6, and C delta. So I've diagrammed that on this page over here. So we can end up with 2 different messenger RNAs. So here we have, what were the numbers here? This one was number 2, this one was 2, this one was 4. And now we removed everything between 4 and C mu right here. And we have a poly A tail. Ooh, I messed that up, didn't I? Let me fix that on the fly here. My eraser. So C delta is still going to be there. But there's a stop codon right here, translation stop codon right there. So when this is translated, we're going to get a heavy chain that looks like this. So translation will go from, say, here to here. And then this is just not used. This is going to give us the variable region here with this constant region. This is the C mu. This is a Greek letter. It looks like a U, but I'm trying to diagram the Greek letter mu. C mu constant region. So it's the heavy chain for the IgM isotype of antibody. And this one right here, which results if this is the 5' splice site and the 3' splice site here is used, will be translated to give us this type of heavy chain, where this is the C delta constant region. And then when this is used in an immunoglobulin, it is the IgD isotype. So that's essentially an overview of VDJ recombination at the IGK, IGL, and IGH loci. What I really focused in on here was just how these different segments are joined together and how they can be transcribed to make and translated to make the light chain and the heavy chain. In the next lecture, what we're going to do is specifically look at the proteins involved in joining the segments together and how they work to join them together very precisely, and how they can introduce, I guess, additional variation between the segments during the joining process that can help increase what we're going to call junctional diversity between the segments and allow the immune system to recognize a very large range of antigens. Okay, so I hope you understood that, and see you in the next video.